Okay. Awesome. So it looks like we are live. Hi, everyone. And it's so awesome to be live with you all and see you all settling into this live stream. It's about 10:58 right now, so we have 2 minutes before we officially get started. But we just want folks to, jump on into the live stream, get settled in for a really, really great conversation that we have lined up for you all. But hey, it's so great to see you guys. And as we're waiting for 11 o'clock to roll around, for those of you tuning in, go ahead and say hi in the live chat and introduce yourselves and say where you're watching from. We love to see where our viewers are tuning in from, and maybe you'll even get a special shout out from myself or Melissa during our chat today. All right. Oh, hello. It's so great to be here with you all this morning. If you're just joining us, we are going to get started in just about a minute. We are giving everyone some time to get settled in, get comfortable, and get ready for this really great Labside chat. Oh, it looks like Zach is watching from DC. Hey, Zach, it's so great to see your comment in there. All right. So it looks like it is 11 o'clock. So let's just go ahead and get started. So hi, everyone, officially, and welcome to a very special Shark Science Week themed Labside chat with the Seymour Marine Discovery Center. My name is Jen Cormier. I'm the Visitor Programs Manager with the Seymour Center, and I'm so happy that all of you are tuning in today. And so today's Labside chat is an opportunity to have a short 20-minute conversation with a scientist from UC Santa Cruz's Long Marine Lab or Institute of Marine Sciences. And in this virtual world, we are so excited to continue to show how scientific research plays a role in the understanding and conservation of the world's oceans. And before we dive into today's incredible chat, I have a big thank you right up top. I want to give a huge shout out to everyone who submitted questions for today's chat in advance. Thank you all so much. There's some really, really good ones, and hopefully Melissa and I will be able to get to all of them today. But for any of you that are tuning in, go ahead and type in your question in the live chat if one happens to come up during the conversation, and hopefully we'll be able to get to those as well. So I'm really looking forward to today's chat because we are speaking with Melissa Cronin. And Melissa is a Ph.D. candidate in the Ecology and Evolutionary Biology department at UC Santa Cruz and is currently working on some really incredible conservation efforts. So Melissa, thank you so much for being here today.
Yeah, thank you so much for having me. I'm honored to be here to talk about this stuff.
Yay, I'm so excited. So we have some really great questions lined up for you. But I said earlier that you are engaged in some really incredible conservation work. So can you tell us a little bit more about that?
Yeah, sure. So of course, as most people know, hopefully it's Shark Week. And a lot of my work doesn't actually focus on sharks specifically, but on a really closely related species group called mobulids. You can see an example of two of them right here. The common name for these species are manta rays and devil rays. So people probably know that more, but if you really want to impress a marine biologist, say mobulids, and you'll be pretty cool, I think. And so what I'm doing at the Conservation Action Lab at UC Santa Cruz, working with Don Kroll, is studying the populations of mobulids using a couple different techniques, and I can talk more about that. And we're working to understand and provide solutions for one of their biggest threats. And that threat is from fisheries bycatch, so the unintentional capture in fishing gear. And this is a really big problem for mobulids, for manta rays and devil rays. And so I'm working on both understanding the problem, but then working with the fishermen to try to develop ways that they can reduce the threat. And I could talk much more about that, but there's a lot of different things they can do to sort of avoid manta rays or to prevent them from being killed in their nets. And so we're kind of trying to work collaboratively to develop some of these solutions and implement them on board the fisheries boats.
Wow, that's incredible. And so I actually want to back up a little bit when you said that rays are super closely related to sharks. And so that is something that's always fascinated me. I know that fascinates a lot of our Seymour Center visitors and supporters. And so can you elaborate a little bit more about how sharks and rays are related? How are they similar? And how are they different?
Yeah, sure. So sharks and rays are part of a larger group called chondrichthyans, and these are cartilaginous fishes. So they, instead of bones like we have, they actually have skeletons made of cartilage. And that is a group that includes both sharks, skates, rays, sawfish, which are pretty cool fishes. And the group that is specifically for sharks and rays is referred to as elasmobranchs. So another cool, impressive word to throw around is elasmobranchs. And these two, they're actually quite similar in sort of morphology, in biological characteristics, and some of these, and of course in some of their threats. Sharks are also pretty threatened by bycatch, many species of sharks. And so there's obviously some differences. Sometimes I like to refer to rays as flat sharks or pancake sharks. I've heard someone call them flappy sharks. But in all scientific seriousness, they are closely related, which is why, during Shark Week, it's a great time to learn about these species that are, sometimes less, maybe receive less public attention in the media, but are really incredible, have really incredible characteristics and are seriously threatened. So I like to think of it as a Elasmobranch Week rather than just Shark Week.
That's great. So Melissa, you gave us two new words to add to our vocabulary. So elasmobranchs and mobulids. So I better write those down.
Oh, yeah.
Awesome. And so you said that you are investigating strategies to help reduce the bycatch of these species. Can you talk a little bit more about like what bycatch is and why does that have such a huge negative impact on the species that you study?
Yeah, absolutely. So essentially, bycatch is when you're fishing for one thing and you catch something else. It's really that simple. And it happens in pretty much every fishery or nearly all fisheries around the world. The context in which I look at bycatch is in our global tuna fishing fleet. And this is a fleet that uses a couple different gears, but it captures a lot of mobulids, about 13,000 a year at least. And so this is a real impact on mobulid populations, which are already in decline around the world from both bycatch and also from targeted fishing. So it's a serious threat. It's also a series of fisheries. The tuna fisheries are centrally governed by very, very large bodies, so management bodies. And that means that if you can enact some sort of changes to fishing at that level of a centrally governed tuna fishery, it's very, very large, you can have really big scale impact. And so that's why I'm interested in this kind of large tuna fishery setting. And these vessels operate mostly on the high seas. So they're kind of way out in the middle of nowhere. They use huge nets. Their nets, I just recently calculated, the size of the net is the equivalent to 16 football fields across. So the scale is almost like too enormous to imagine. And of course, if a mobulid, a manta or a devil ray is swimming along, it's just going to get sort of scooped up in that net. And so there are a few things that we're doing in the lab. The first part is a population genetics study. And so the way we're doing that is we're collecting very small tissue samples of the mobulids that are caught on board. So they're out in the middle of the ocean fishing for tuna, and a mobulid comes up in their net. The observer, who we've trained, will take a very small piece of the tail. They have really long tails, so it's almost nothing. And they put it in a vial and they send it back to me in the lab for analysis. And what I'm doing with some really wonderful research assistants here at Santa Cruz is trying to understand from those teeny tail samples, we can understand which species are getting caught the most. So who's getting caught? There are several species and we want to know which ones are getting hit hardest. And then of those species, we want to know which sort of populations within those species are getting caught. And whether, importantly, this is the important question, is whether there are smaller individual populations in that species that might be getting hit really hard, or whether there is one large population that is genetically similar. And that is a really crucial metric for conservation to guide conservation efforts, because you would do very different things depending on if you have small populations or one large population, which kind of has this potential for, well, it just has different strategies. So that's the first side of things. That's kind of like the really hard sciencey side of things with a lot of lab work and pipetting. And that will give us a whole lot of information that we can actually deliver to the tuna fisheries to say, hey, here's the sort of impact of this bycatch. The other side of things that's a more applied project that we're working on just recently is to try to develop different ways that they can fish and handle the animals that they capture so that they're less likely to die from bycatch. And so what we've done is organize a series of workshops, pretty simple, pretty like low tech, approach, but we're bringing skippers. So those are the captains of the vessels, crew, and then fisheries observers, who are the people that go on board and collect data. And we're hosting a series of workshops in Ecuador this coming year, hopefully, given the uncertainty in the world right now. But we're having them work with scientists and fisheries managers to change the gear that they use. So kind of these slight modifications, anywhere from changing the way that they handle the animal as they release it, or to actually implementing sort of larger scale hydraulic systems on the vessels. And then the plan is to pilot one of these sort of innovations, we're calling them, on board the tuna vessel. And so it's a bit of a long process from kind of thinking of the idea to the eventual implementation. But because we've been working with the tuna fishery every step of the way, both in the genetics work, in the sort of design of these workshops, and eventually in the implementation of the sort of solutions, there's actually a really great opportunity for it to sort of scale up over a fishery that, in our case, extends all the way out, past Galapagos Islands, the whole Eastern Pacific Ocean. And so if you can have impact on those vessels, it's really an ocean basin scale impact, potentially. So those are the two sort of sides of things. There's the genetic question. We want to know what is the nature of the impact. And then the conservation side is this collaborative process of developing solutions.
Definitely. Well, it sounds like every single step of the way that you're doing to help protect these species is just so collaborative, and that just seems so important to your work. And so I have to ask, so you said on the genetic side of things that you take a little small sample of the tail. So how many little samples or tail samples do you have in your lab right now, just out of curiosity?
So, oh God, I'd have to look at the freezer, which is kind of scary. We have, I believe, over 600 tail samples. Some of them are just tissue samples, like, pectoral fin samples, which is a lot. They're from all over the world. So my focus is on the Eastern Pacific, but we're also interested in sort of comparing other areas as well. And so we have samples from the Philippines, from India, Mexico, Peru. We have some from West Africa. And it's really cool because we're going to be able to look across, an ocean basin to see, is there a difference in these individuals from the Philippines to Ecuador? That's a pretty amazing scale. And nobody has really done that work yet. Somebody told me, this is hearsay, but somebody told me, and this is a reputable source who's a really well-known manta researcher. He thinks that we have the largest collection of mobulid tissue samples in the world, which is a very specific claim to fame, but one nevertheless.
Exactly. Yeah. If that's true, I mean, I would, brag about that all the time. That's incredible. And so you said another part of the conservation work that you're doing, right? We have the genetic side, but then we also have the very collaborative side where these animals are potentially caught as bycatch in these nets. And so these animals are pretty big, right? And so you said that you're developing tactics to re-release these animals back into the ocean so they don't suffer any negative effects. So like, just how big are these animals?
They are huge. Yeah, I think of it, they're one of those animals that people just totally underestimate the size. So the comparison I like is, and people may have heard this before, they've heard me talk about these, but they can reach 2 tons, which is about the weight of a Honda Civic. So very, very, very heavy. And those are just the largest ones. And then they can reach the width, so wingtip to wingtip, horizontally, as a giraffe is tall. So these are big animals. You know, when we dive under them, they blot out the sun above us, which is sometimes pretty spooky, but really cool. And so, you know, as big and beautiful and kind of majestic as they are, it actually is a huge problem for them in terms of bycatch, because they're so darn heavy. So when the tuna fishery or even another type of gear is trying to deal with them, one of the biggest problems is for the large ones, they just actually can't pick it up physically. When they take samples for us, they take a video and photos, and we'll see them sort of, sometimes the poor crew is kind of struggling under the weight of the animal to get it overboard. And it causes problems because they can just sit on deck and sort of their organs will become crushed if they're sitting there too long, not buoyed by the water. And so what we've seen, which is really cool so far, we've seen in these videos, is that they've actually started to develop sort of homemade like solutions on board that are potentially really promising. So there's a couple things like a grid, a sort of modified net, and I won't go into the technical details, but we've seen these like innovations coming from the fishermen and the crew themselves, which is really exciting to see. And that's kind of what we're after with these workshops, is really formalizing and developing those and testing them. And so, you know, it's not easy. It's not easy to get a Honda Civic off of your deck, but there are certainly solutions. And on a boat, a really large vessel, there's, you know, the hydraulic power to do it, frankly. Those are the really big ones. These ones are much smaller, and there are a couple species that are generally pretty small. They're still kind of heavy when you pick them up, but you can, you know, toss them overboard, or we can handle them a little more easily without, you know, a hydraulic crane.
Wow, that is incredible. I drive a Honda Civic, so I'm pretty much driving around in a manta ray.
You are a giant oceanic manta ray. It's your car.
Wow, that's incredible. All right. Well, I'm telling you, that's going to be my new fun fact that I tell at parties and conferences from now on.
Oh, cool.
All right. So, Melissa, we got a question, and it's actually really interesting and actually relates to your background, so I'm excited to ask this. So rays do these incredible behaviors, such as jumping out of water or gathering in these large congregations. And so have scientists figured out why they potentially do these types of behaviors?
Oh yeah, it's a really good question. Yeah, so the species that this is called the monk's devil ray, Mobila monkiana. And that's a species found on the coast of Central and South America, on the West Coast. They are known for both of those two behaviors, the amazing sort of balletic jumping, and then these aggregations that, I mean, just Google like manta or devil ray aggregations, and you'll see some pretty awesome videos of hundreds, thousands, even potentially more individuals all schooled in one. And it's really, really incredible. There has not been a lot of work on those two behaviors. However, our lab, and Dawn and Kelly Ziliakis, our lab manager, they've been studying this species for more than 10 years now. And we've gone down there. I've gone down there a couple of times to their study site in Baja, and they're totally incredible, incredible animals. I mean, you can just be sort of going along in your small boat in your panga and hear this like sort of slap, 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 slap. And you'll look over and see a bunch of devil rays bouncing on the water. Or, you know, you'll see these amazing groupings of them in hundreds, thousands. And so as to the question of why, there is not like a reliable scientific answer for this. For the jumping, the best hypothesis that I've heard, and the one that I sort of am inclined to trust most, is that it's for communication. and that there's some sort of utility of making a sound on the top of the water that sort of reverberates into the water column that indicates to another individual, hey, I'm here. It could potentially mean there's a lot of food right here. It could mean come over here because we're avoiding a predator together and want to school up. I mean, there's a lot of different, reasons to communicate, but that seems like the best one, which to me is amazing because it's a real indicator of sort of social and potentially cognitive ability. And for these animals in particular, they have the potential for higher than expected cognitive abilities. So this group has the largest brain to body ratio of any fish, meaning that their brains are very, very large relative to their body size. And they also have highly developed parts of their brains that are associated with complex problem solving and with social behavior. And so all these things kind of indicate these, like the brain physiology or morphology and the these sort of social behaviors, complex social behaviors, indicate that they are an intelligent species or more intelligent than you might assume from a fish. And they're really, really social. They have really complex courtship and mating behaviors, sort of vortex feeding behaviors where they use the water, or they use the sort of behavior to maximize the efficiency of feeding. So they're really cool, really interesting species, but relatively not well studied. So if there's anybody there considering a dissertation topic, that is a great question.
That's awesome. Well, thank you so much for giving us insight into that. And so, Melissa, I only have 2 more questions for you. And my next question is, how did you decide that this conservation path was the one for you and that you wanted to work with these types of animals? Was there just like a moment where a light bulb went off and you're like, you know what, shark, rays, these are my jam. I love them. And this is what I want to study. How did you decide that this is what you wanted to do?
Oh, yeah. Well, my background was unusual and not a straight path, certainly. Perhaps the opposite. So I did my undergrad in journalism and environmental studies, and then I was an environmental journalist for 3-4 years in New York. And I was mostly interested in covering environmental issues. So I was covering stuff like methane emissions, environmental politics and law. I was covering conservation, sort of, trophy hunting stories. And in particular, I was covering some shark fin trading routes. And it was a really amazing career, totally fascinating work. And one of the cool things that when I was covering this shark fin issue, which is a really complicated issue, but I was just sort of covering a very small part of it. And we had noticed that these large shippers, so UPS and FedEx, were shipping shark fin shipments in like commercial airplanes in the cargo. And we're like, oh, wow, that, maybe passengers would want to know that. And so I wrote a story about it, and immediately a boycott was sort of called. And even the threat of the boycott for such a large company is probably not that great, but even that threat was enough to make them change their regulations immediately and stop shipping shark fins. So it was amazing. I was like, wow, this is like a concrete victory just happened. There's a lot more to that story and how it all like falls out, but it was really exciting. And that really made me think, okay, I want to, go into actual conservation. I don't want to write about conservation. I actually just want to do it. And then also I was covering the Trump election or the campaign, and that was horrible. So I wanted to leave journalism. So I was super, super fortunate to, be accepted into the lab at Santa Cruz. I'm, not a conventional candidate, certainly, for grad school, but it worked out amazingly. It also wasn't a easy, I didn't immediately choose this project. It was, I'm sure, not, simple, but because our lab has worked on this species, the Monk's devil ray in Baja California, it was a natural extension to look at this species group, but in a different setting. And so, they have done all this like really foundational ecology, conservation of mobilids And now we're kind of moving into this tuna realm, which is a much grander scale, and a sort of a different problem, but kind of builds on that great foundation that the lab already has.
Yeah, that is so incredible to hear about your background and how you got to where you are today. And what I can say is that the folks over at the Doris Duke Conservation Scholars Program are saying that UCSC is super fortunate to have you, and I couldn't agree with them anymore. So that's wonderful. Okay, so my last question for you, and a question that I have to ask every single scientist, is what is the best part of your job?
Oh gosh, that's a good question. Okay. I was thinking about just doing like lab work and how tedious that can be, but because I was just doing it. No, I think the best part is working with fishermen and fisheries observers. It has been really eye-opening, especially as a conservationist, coming from a developed country, trying to work on a sort of charismatic species. It's been really eye-opening to learn about the, interests, the considerations, and the things that people need in order to make conservation happen. So there's two groups that I have sort of worked with in one in smaller capacity than the other, but in Baja, the research site, the field site is a small island, and there's an amazing group of fishermen who lived there for many, many years, or many generations. And working with them was really fascinating. And the cool thing is that Santa Cruz has a long history of taking classes down to that site. So there's a lot of real value there. And then the other group is this large scale fishery, which, we've gone to Ecuador to sort of train people to take samples. And now we're surveying the fishermen and the skippers. And it's been really interesting to see, that's a really large fishery with different economic structure than, say, a small scale fishery in Baja in Mexico. And to understand the reasons why they will pursue conservation action, which are very different. So in the case of some large tuna fisheries, it's because they want a sustainability certification. And, that's a complicated issue as well, but you can really capitalize on that if you want to get something done for a threatened or endangered species. So that's been really cool. I mean, it's also just been cool to be in the field with the animals. They are so beautiful and fascinating. I'll just share one last story of being in Baja last year, and it was a full moon, and the mobulas, they like the full moon because they can see their prey better, the zooplankton. And so they're generally really active during the full moon. And I was sleeping outside with a collaborator, and I remember we just heard this slapping in the middle of the night, all night long. It's actually a little loud. And it was the mobulas jumping in the dark. And every once in a while you can just glimpse a little, one of them in the moonlight. And that was just such a special experience, kind of being close enough to hear them doing this really fascinating, mysterious behavior was cool. So yeah, interacting with the fishermen and hearing their kind of accounts. Now some of them will send me updates from the middle of the ocean using their satellite phone and say, "Oh, we caught a mobula today. Aren't you excited?" And that's been pretty unique experience with my PhD.
That's so awesome to hear, Melissa. What special memories that you have. And thank you so much for sharing that with us. And I just really can't thank you enough for sharing your expertise and being here to talk about your conservation work. It's so inspiring what you do. And we are so lucky, that we have you working on this issue. So thank you again. And, I can keep asking you questions probably for the next couple days to learn more about this topic, but unfortunately we are just out of time. So do you have a website or a place online where folks can go to learn more about you and your research?
Yes, absolutely. My website is called melcronin.com, MEL, and that has some information about the stuff I'm working on. My Twitter, you could follow me on Twitter. It's at Melissa_Cronin. I believe that, yes. And if folks have questions, I'm always happy to answer them and nerd out together on Twitter about, mobula stuff. And yeah, I'm so grateful to have been here. Thank you so much for hosting me, Jen, and to the Seymour Center, which has been so amazing to me and hosted me, talking about manta rays many times. And thanks to Doris Duke too for the shout out.
Awesome. Well, I will make sure to put those links in the video description below so folks can access them later. But I can tell you that Melissa's Twitter is the place to be. So ask her lots of questions. So great. I also want to send a really big thank you to everyone that has tuned in this morning. It's really supporters like you that make these online programs possible, such as Labside chats or our virtual Shark Science Week possible. So if you're in the position, I ask that you please consider making a donation or becoming a member with the Seymour Center. You can find more information on our website, which is seymourcenter.ucsc.edu, or you can go ahead and click in the links below in the video description. And keep celebrating Shark Science Week with us all week long. We're celebrating all the way till Saturday. So there's lots of different activities and videos and crafts that you can find on our website. Again, all of those links are in the video description below. But thank you again, everyone, for tuning in. Happy Shark Science Week, and we will see you next time. Bye, everyone.